Welcome again to June Summit Biochemistry Hub, your go-to place for mastering biochemical concepts one step at a time. I remain your host, Dr. Juliet. Today we shall be looking at methionine metabolism under amino acid or protein chemistry. Methionine, as we know, is an essential amino acid and so must be obtained from our diet. Methionine is important in protein synthesis, being the first amino acid added during translation process. Methionine has various interconnected pathway and processes. Let's see that quickly. Methionine originally is converted to S-adenosylmethionine, the molecule that we know to be notorious for methylation reactions that helps in epigenetic control. That reaction, catalyzed by methionine adenosine transferase, uses ATP and releases inorganic phosphate with the product S-adenosylmethionine, which we call SAM. SAM is involved in the methylation of DNA, histones, and proteins, thereby taking control of genetic expression. Now, this S-adenosylmethionine can be demethylated by methyltransferase to produce S-adenosylhomocysteine. When this happens, it can also be converted to homocysteine by S-adenosylhomocysteine hydrolase or homocysteines, releasing adenosine to return to homocysteine. Now, homocysteine can go through various pathways. It can either be remethylated to give methionine, or it can go through the transsulfuration pathway. So either it takes the methionine cycle pathway to be converted back to methionine, or it enters the transsulfuration pathway to produce glutathione, a tripeptide molecule that is notorious as an antioxidant molecule. Now, how do we salvage methionine from this? There are two ways. It can either go through the choline-glycine cycle or through the folate pathway. Now, if it's going to be returned back to methionine by the folate cycle, what it needs to react with is 5-methyl tetrahydrofolate. And that reaction involves vitamin B12 to give tetrahydrofolate and methionine, catalyzed by the enzyme methionine synthase. What happens when there is deficiency of vitamin B12? If there is deficiency of this coenzyme, this reaction cannot go forward. And so we're going to have accumulation of 5-methyl tetrahydrofolate, the metabolic condition we call folate trap syndrome. Due to deficiency of vitamin B12, this reaction cannot go forward, so we have insufficiency of methionine, and then we have accumulation of homocysteine and folate trapped in this form, which we call folate trap syndrome. Now, also, methionine can be salvaged from homocysteine via another cycle, the choline-glycine cycle, where choline is converted to betaine, which remethylates homocysteine to give us our methionine with the production of dimethylglycine. This can continue this way to give us glycine. So this cycle is simply conversion of choline to glycine, which also helps in the methylation of homocysteine to methionine. Why is it important? It helps to salvage this. It also helps to produce glycine, which is needed through this pathway for the synthesis of glutathione right here. Now, in the transsulfuration pathway, we see homocysteine being converted to cystathionine by cystathionine beta synthase. Vitamin B6 is essential for that reaction. Cystathionine is converted to cysteine, another amino acid essential for protein synthesis, by cystathionine gamma lyase. Vitamin B6 is also essential for that reaction. Cysteine reacts with glutamate to give gamma glutamyl cysteine, and then glycine comes in to give glutathione. So we see that this cycle, choline-glycine cycle, not only helps to remethylate homocysteine to give them, but also provides glycine for glutathione synthesis via the transsulfuration pathway. I trust that we enjoyed ourselves looking at this day. I encourage you to continue to follow, like, share our video, and subscribe to our channel. Until I see you again It is bye-bye for now.